Genetics is the study of heredity and variation. Heredity means the inheritance of characters from generation to generation is called heredity. When genes occur in more than one form, we call it as multiple allelism, and the classical example for occurrence of multiple allelism is ABO blood groups. Punnett checkerboard concept was proposed by Reginald C. Punnett, and the checkerboard is used to show the genotype and phenotype of the cross between the two individuals. Hello everyone, welcome back to yet another revision class of 2nd PUC chapter 5 called Principles of Inheritance and Variation. Myself B. Shobharani, faculty from the Department of Biology, Vidhyaashram Pre-University College, the Temple of Excellence. So let us move on to discuss about a very important chapter called Principles of Inheritance and Variation, which is very important from examination point of view as it carries a very big marks. That is, the weightage of marks for this particular chapter is 10 marks. So this chapter has lot of weightage from examination point of view. So let us discuss as to what are the important concepts you will have to concentrate on for writing the answers for this particular chapter called Principles of Inheritance and Variation. As I said, the weightage of marks for this particular chapter is 10 marks. What are the type of questions that are asked for 10 marks? Like for example, 2-marker question you may get 1 question, and for 3-marker question you may get 1 question. So there it is 5 marks, and 5-marker question 1 question, it is 5 marks. So total 5 plus 5 is equal to 10 marks. So you may get 2-marker question 1, 3-marker question 1, and 5-marker question, that is 1 question. But again, as I said, in 5-marker question you may get a split question for 2 plus 2 plus 1, or you may get for 3 plus 2. So that is how the questions are asked in the board examination. So the weightage of marks is 10 marks, 2-marker question 1, 3-marker question 1, and 5-marker question 1. So let us go as to what is the synopsis part of this particular chapter. So we all know that gene is nothing but the unit of heredity. So in this particular chapter, we are going to study about principles of inheritance and variation, where we have to learn about as to what is gene. As I said, it is a unit of heredity, or gene is a segment of DNA. And as we all know that Gregor John Mendel, who is called the father of genetics, was the man who laid down the foundation for the study of genetics, and he worked on the garden pea plant for 7 long years, and he was able to get 7 contrasting characters, and his experiments are called as what? Hybridization experiments. Hybridization experiments. And we all know that Mendel's work was brought into light only after his death by 3 very important scientists, that is Hugo de Vries, Tisha Mark, and Karl Correns. So these 3 scientists, they brought the Mendel's work into light, and they formulated the ideas of John Gregor Mendel in the form of laws called Mendel's laws of heredity. Now let us understand what is genetics. As I said, actually John Gregor Mendel was of the opinion that there is something which passes on stably, remaining unchanged from the parents to the offspring. So he called that something as factors. He called them as what? Factors. But today we call them as what? Genes. We call them as gene. So as I said, gene is nothing but a segment of DNA, or it is the unit of heredity. So what is that branch of science which deals with the study of gene? It's heredity. So genetics is the study of heredity and variation. What is heredity then? Heredity is nothing but inheritance of character, passing on the characters from the parents to the children from generations to generation is called heredity. So what is genetics? Genetics is the study of heredity and variation. Heredity means the inheritance of characters from generation to generation is called heredity. Then what is variation? The change in the hereditary characters is called variation. And as we all know that variation is possible only through sexual reproduction, and variation leads to evolution. So variation means the change in the hereditary characters is called variation. So what are the variations? Then as I said, variations are the raw materials for evolution. Then what are we going to study in this particular chapter? Mendel's laws of inheritance. As I said, whatever ideas he formulated from the study on the garden pea, he was not able to formulate, but his ideas were formulated in the form of laws called Mendel's laws of inheritance, where we study about inheritance of one gene. Inheritance of one gene pertaining to law of dominance and law of segregation. Right. Then non-Mendelian inheritance, like we study about incomplete dominance, where we take a classical example of the dog flower or the snapdragon or the antihyrum, the 4 o'clock plant or Mirabilis jalapa. Co-dominance, as we know, the very classical example for co-dominance is AB blood group. So multiple allelism, when genes occur in more than one form, we call it as multiple allelism, and the classical example for occurrence of multiple allelism is ABO blood groups. Then we'll be studying about pleiotropy also. Then inheritance of two gene is verified by Mendel's dihybrid cross. As we know, the previous slide, as I said, Mendel's law of inheritance, that is inheritance of one gene, is studied by Mendel's monohybrid cross. So if at all they ask, explain the inheritance of one gene, then we have to explain about monohybrid cross. If at all they ask for inheritance of two genes, then we have to explain about dihybrid cross. Law of independent assortment is verified by Mendel's dihybrid cross. Then you'll be studying about chromosomal theory of inheritance, which was put across by Walter Sutton and Boveri proposed the chromosomal theory of inheritance, and it was experimentally proved by T.H. Morgan, Thomas Hunt Morgan, who worked on Drosophila melanogaster, which is called the Cinderella of genetics. Then that's what the experiment by Sutton and Boveri, chromosomal theory of inheritance put across by T.H. Morgan, who worked on Drosophila melanogaster, which is called the Cinderella of genetics. So if at all they ask for inheritance of two genes, then you have to explain about dihybrid cross. Dihybrid cross, that is law of independent assortment, can be proved by dihybrid cross. Then chromosomal theory of inheritance, which was put across by Walter Sutton and Theodore Boveri, and chromosomal theory of inheritance was experimentally proved by T.H. Thomas Hunt Morgan, who worked on Drosophila melanogaster, which was called as Cinderella of genetic linkage and recombination. Then you will be studying about sex-linked genes, then polygenic inheritance and gene mapping. Then sex determination, you will be studying about sex determination in human, then sex determination in honeybee, then pleiotropy, mutation, that is a sudden heritable gene, change in the gene, then the causes of mutation. Then you'll be studying about pedigree analysis, which is very much required to identify the abnormalities in the gene. Then genetic disorders like Mendelian disorders and chromosomal disorders. Chromosomal disorders like you can come across the Klinefelter syndrome, Down syndrome, Turner syndrome, all that you'll be studying in this particular concept called chromosomal disorders. Now, let us move on to the 2-marker question. So, what will be the type of questions which are asked for 2-marker questions? First question, who discovered Mendel's work? As I said, John Gregor Mendel, who is rightly called as father of genetics, worked on pea plant for 7 long years, and all the ideas that were formulated by John Gregor Mendel were put across in the form of laws called Mendel's laws of heredity, and John Gregor Mendel's work was brought into light by 3 very important scientists by name Hugo de Vries of Netherlands or Holland, then Karl Correns of Germany, and Erich von Tischenmack from Austria. So who discovered Mendel's work? Hugo de Vries, Karl Correns and Erich von Tischenmack. Then what is Punnett checkerboard? Who developed it? Punnett checkerboard concept was proposed by Reginald C. Punnett, and the checkerboard is used to show the genotype and phenotype of the cross between the two individuals. So what is Punnett? Like when we write the checkerboard like this, where we take the male gametes at one side and the female gametes on one side. So you can see the Punnett checkerboard concept was proposed by Reginald C. Punnett, hence the name Punnett checkerboard. So you put a board like this and write. The checkerboard is used to show the genotype and phenotype of the cross between the two individuals. Then what is linkage? Mention its significance. So what is linkage? It is a tendency of 2 or more genes to remain together in the original combination on the same chromosome from generation to generation. So what is linkage? It is the tendency of 2 or more genes to remain together in the original combination on the same chromosome from generation to generation. So what is the significance of linkage? Helps in the progeny to have parental gene combinations. So what is it helping in? Helps in the progeny to have parental gene combinations. So next question is what? What is pedigree analysis? Mention its significance. So what is pedigree analysis? It is an analysis of inheritance of traits or characters for several generations in a family is called the pedigree analysis. So we all know what is the significance, what is the importance of knowing the pedigree analysis. In human genetics, pedigree study provides a strong tool which is utilized to trace the inheritance of a specific trait, abnormality or a disease. So what is pedigree analysis and mention its significance? It is an analysis of inheritance of traits or characters for several generations in a family is called pedigree analysis. Significance in human genetics, pedigree study provides a strong tool which is utilized to trace the inheritance of a specific trait, abnormality or a disease. Moving on to the 3-marker question. What is point mutation? And give one example each. So point mutation. What do you mean by point mutation? Mutation is sudden heritable change. But what is point mutation? Point mutation refers to the mutation arising due to a change in a single base pair of a DNA. So in that, hence the name point mutation. So point mutation refers to the mutation arising due to a change in a single base pair of DNA. What is the classical example we are going to give for point mutation? It is sickle cell anemia. What is the sickle cell anemia? The defect is caused by a single base substitution at the 6th codon, that is GAG to GUG, of the gene coding for beta globin chain. So consequently what will happen? The amino acid glutamic acid is substituted by valine at the 6th position of the beta globin protein. So what is point mutation? And give an example each. Means point mutation refers to the mutation arising due to a change in the single base pair of DNA. Example, sickle cell anemia. What is this defect? The defect is caused by a single base substitution at the 6th codon of the gene coding for a beta globin chain. Consequently, amino acid glutamic acid is substituted by valine at the 6th position of the beta globin protein. Next question. Explain codominance with the help of one example. What is codominance? The very word itself will tell you. Codominance is the phenomenon in which two alleles of a gene are equally dominant and express themselves in the presence of the other when they are together in an individual. So codominance is the phenomenon in which two alleles of a gene are equally dominant and express themselves in the presence of the other when they are together in an individual. I think all of us, example, occurrence of AB blood group. AB blood group, you can give an example of codominance. Codominance. So here, blood group of AB in this, the allele IA and IB are codominant and both express themselves producing the glycoproteins A and B, making the blood group AB. That is what explain codominance with help of one example. Then work out a cross between true breeding red and white flowered dog flower plant, that is snapdragon, up to F2 generation. So when we take the parents, that is red flowered plant is crossed with a white flowered plant. So what will be the gametes? Capital R, small r. So when you cross the generators, these are the gametes. Capital R, small r. So what will be the F1 progeny? F1 means the first filial generation will be pink flowered. So this is an example for incomplete dominance. So when there is a cross between a dominant and a recessive, the dominant character fails to express itself completely in the F1 generation and there is an intermediate character appearing in the F1 generation. So here instead of red appearing, which is the dominant character, we find an intermediate color character appearing, that is pink flower. So when a red colored flower is crossed with a white flowered plant, you get F1 as all the flowers will be pink flowers. So self-pollination, that is what do you mean by cross-pollination? That is self-pollination. That is you get F2 by F1 into F1. So this is F1. So cross this. So what will be the gametes for this? Capital R, small r into capital R, small r. So as I said, when a red colored flower is crossed with a white color flower, so what will be the gametes? Capital R, small r. So what will be the F1 generation? that will be the pink color. As we know, incomplete dominance is where the dominant character will not completely express in the F1 generation, it will not completely suppress the recessive character. Instead, an intermediate character arises, and that is called pink flower. So self-pollination is, that is F2 you get it through self-pollination, F1 into F1. So what will be the gametes here? Capital R, small r, capital R into small r. So going for F2 generation, F1 into F1, that is capital R small r into capital R small r. So that is capital R small r, capital R small r. So when you cross RR, capital R, capital R, capital R small r, capital R small r and small r small r. So how many you get red flowered? 1. How many you get pink colored? 2. How many you get white flowered? 1. So red flowered is 1, pink flower is 2 and white flower is 1. So what is the ratio? 1:2:1 is the phenotypic ratio. So here it will be both the phenotypic ratio and genotypic ratio is 1:2:1. State and explain the law by taking inheritance of one gene, that is the law of dominance and inheritance of one gene. As I already said, that is law of dominance and inheritance of one gene. So law of dominance, characters are controlled by discrete units called factors. Factors occur in pairs. In a dissimilar pair of factors, one member of the pair dominates, that is the dominant gene, and the other is recessive. As I said, the inheritance of one gene is verified by Mendel's monohybrid cross. Monohybrid cross, or it is also, that is the law of segregation is verified by Mendel's monohybrid cross. So what is the inheritance? The law of segregation, which is also called purity of gametes or monohybrid cross. What is the statement? The factors or alleles of a pair segregate from each other without blending such that a gamete receives only one of the two factors. So when you cross tall pea plant with a dwarf pea plant, so in the F1 generation you get a hybrid tall. So when you go for F2 generation, that is selfing F1 into F2. So based on that, let us see the schematic representation. So here schematic represent parent generation, that is homozygous tall pea plant and homozygous pure dwarf pea plant. So what will be the phenotype is tall pea plant crossed with a dwarf pea plant. So what will be the phenotype? Capital T, that is tall, then small t, small t. So what will be the gametes? Capital T and small t. So when you cross them, you get F1 generation, that is capital T, small t, which is a hybrid tall. So when you go for the first filial generation, that is capital T, small t into capital T, small t. That is capital T, small t into capital T, small t. So heterozygous tall hybrid. When two F1 hybrid offsprings are self-crossed, that is capital T small t into capital T small t. We can go for a checkerboard here. So Punnett square showing gametic combination. So you take the gametes on one side, capital T small t, capital T small t. So here you get homozygous tall. When you cross capital T with capital T, you get homozygous tall. Capital T small t, you get heterozygous tall. Then capital T small t, again you get a heterozygous tall. Small t you get, that is small t small, homozygous dwarf you get homozygous dwarf. So varieties of plants in F2 generation is what? Tall pea plants, how many you get? You get 3. Dwarf pea plants you get 1. So here, so we get capital T small t, capital T small t and small t small t. So this is, so 1 you get pure tall, 1 pure tall, 2 hybrid tall, then 1 pure dwarf. But when you take totally 3 tall plant, when you take the phenotypic ratio, 3 tall is to 1 dwarf. So that is what? So what will be the phenotypic ratio? Phenotypic ratio 3:1. What will be the genotypic ratio? Genotypic ratio will be 1:2:1. 3 tall is to 1 dwarf, or 1 pure tall, that is 1 capital T, 2 capital T small t and 1 small t small t. So this is the phenotypic ratio or genotypic ratio of one gene inheritance. That is what, so tall, heterozygous tall, 1:2:1. Crossing again, you get what? Test cross. What is test cross? Crossing F1 into parental recessive. Why do we go for a test cross to verify hybrid, that is the F1 generation is homozygous or heterozygous. So how do we get test cross crossing F1 into parental recessive? That is capital T small t into small t small t. Again you go for a checkerboard where you cross here, you get that is capital T small t crossing with small t small t. So what do you get? Capital T small t small t small t capital T small t small t small t. So 2 you get hybrid tall and 2 you get dwarf. So 2:2 or 1:1. So this kind of you will come across in, that is in one gene inheritance, taking a tall pea plant crossed with a dwarf pea plant. Next question. What is polygenetic inheritance? Give an example. A kind of inheritance in which a trait is produced from the cumulative effects of many genes. Inheritance of human eye color, skin color, height and weight in human beings is a classical example of polygenetic inheritance. So what is polygenetic inheritance? A kind of inheritance in which a trait is produced from the cumulative effects of many genes. So human skin color is another example for polygenic inheritance. So in a polygenic trait, the phenotype reflects the contribution of each allele. To understand this better, let us assume the 3 genes, that is A, B and C, which control skin color with the dominant forms called A, B and C responsible for dark skin color. So the recessive forms will be a, small b and small c for light skin color. So the genotype with the dominant alleles will be what? AA, BB, CC will have the darkest skin color. So all the recessive alleles, for example, you take AA, BB, CC will have the lightest skin color. So the genotype with 3 dominant alleles and 3 recessive alleles will have an intermediate skin color. So in this manner, the number of each type of alleles in the genotype would determine the darkness or lightness of the skin in an individual skin. Then going again for a 5-marker question, what are multiple alleles? Give an example. This is a split question. Then write the symptoms of Klinefelter. So it is for 2 plus 3. So what are multiple alleles? Give an example. Write the symptoms of Klinefelter syndrome. Answer. What are multiple alleles? More than 2 alternative forms of a single gene occupying the same locus on the homologous chromosome and control a single character is called multiple alleles. That is, what are multiple alleles? Give an example. Write the symptoms of Klinefelter syndrome. That is, more than 2 alternative forms of a single gene occupying the same locus on the homologous chromosome and control a single character is called multiple alleles. So blood group in man is a classical example for occurrence of ABO blood group in man is a classical example for multiple allelism. Symptoms of Klinefelter syndrome. This is an allosomal abnormality due to the addition of an extra X chromosome. So this symptoms of Klinefelter syndrome is, this is an allosomal abnormality due to addition of an extra X chromosome. So this is, you can see only in male. So in male we have 44 plus 44. What will be the chromosomal complement? 44 plus XY, that is equal to 46. But in case of Klinefelter syndrome, it will be 44 plus XXY. So there is an addition of one extra X chromosome and that will be how much? 47. So it will be 47. So hence it is a trisomy. So it is due to 2n plus 1, which is called trisomy condition, that is 2n plus 1. So the affected individual is having 47 chromosomes instead of having 46 chromosomes. So Klinefelter syndrome is an allosomal disorder where there is one extra X chromosome to the normal set of sex chromosome. But here you can see that it is due to trisomy. So instead of 46 chromosome, the male will have 47 chromosome where they have one extra X chromosome. So what will be the symptoms of Klinefelter syndrome? They are tall statured, the body shows obesity, scanty hairs on the body, then they show tendency towards female characters like enlargement of breast, which is called gynecomastia, then underdeveloped testes, which is called hypogonadism, and low production of androgens, that is a male hormone, and there is no production of sperms, hence they are sterile males. So all these are the symptoms of the Klinefelter syndrome. Let us go for the recap. Principles of inheritance and variation. As I said, this is a very important chapter from examination point of view, which carries 10 marks. So Mendel's law are law of dominance, law of independent assortment and law of segregation. So these three are, that is all the ideas formulated, put across by Mendel was formulated in the form of laws called Mendelian, laws of heredity called law of dominance, law of independent assortment and law of segregation. Then exception of Mendel's laws are incomplete dominance, where we take an example of snapdragon, codominance, we take then example of blood group, for multiple allelism, ABO blood grouping, then pleiotropy, then polygenic inheritance, where we take human skin color. Then mechanism of sex determination, where we come across male heterogamity and female heterogamity. The examples for male heterogamity is XX, XY type, which we come across in Drosophila melanogaster. Then ZW type, where we come across in birds. XXXO in grasshopper, that is male heterogamity. Female heterogamity ZZZO type. Then genetic disorders, where we classify them into Mendelian disorder and chromosomal disorder. So Mendelian disorders are hemophilia, color blindness, sickle cell anemia, thalassemia and phenylketonuria. Whereas the chromosomal disorder, where the chromosomal number may vary due to either monosomy or trisomy, that is the Down syndrome, Turner syndrome and Klinefelter. So you come across here the Turner syndrome, then here the Down syndrome, then here the Klinefelter syndrome. Then hope you all have understood what type of questions are going to be asked in this particular chapter 5 of your second PUC syllabus called Principles of Inheritance and Variation. Thank you.